My name is Jamie Fontana, and I was mentored by Dr. Wendy McFarlane in my research of monitoring changes in dissolved oxygen, phosphate, nitrate, and pH levels from the upside-down jellyfish Cassiopeia xamanchana under varying photoperiod. Cassiopeia xamanchana are benthic invertebrates found in tropical shallow water. They have endosymbiont algae present in their tissue that allow photosynthesis to take place. This study explored the effects upside-down jellyfish have on nutrient levels present in their environment and their effects when held under different light levels. For our pilot study, the objective was to create an apparatus for continuous monitoring in order to observe the effects of jellyfish present on phosphate and nitrate levels, dissolved oxygen concentration, and pH. This was done by placing 4 jellyfish similar in size into 2 containers and exposing them to the same photoperiod. A third container was used as a control and did not contain any jellyfish. They were then placed inside an incubator set at 22 degrees Celsius. The second focus was determining if there would be a decrease in dissolved oxygen levels and a decrease in nitrate and phosphate levels when in dark conditions, since the Cassiopeia will not be able to photosynthesize. This was done by placing 3 jellyfish of similar densities into 2 containers and having a third container empty as the control. One container was labeled light and was exposed to the normal 12 to 12 photoperiod, whereas the second container was labeled dark because it was not exposed to any light. A non-transparent box was placed over the entire container. The containers were also placed inside an incubator set at 22 degrees Celsius. Using the LabQuest veneer tablets, we were able to record dissolved oxygen and pH levels every 15 minutes throughout the entire 24-hour experiment. Point samples were recorded in triplicates for phosphate and nitrate from each container at time 0 and again after the duration of the experiment. Then a spectrophotometer was able to give a value to the nutrients present. The results of the pilot study showed significant differences between tanks that held jellyfish versus no jellyfish. Dissolved oxygen levels decreased tremendously in treatment tanks 1 and 2, whereas the control remained fairly constant. pH showed a similar trend. Phosphate and nitrate levels increased tremendously in both treatment tanks, and the control had very little change. For the effective photoperiod study, the light container was shown to increase dissolved oxygen levels when the incubator light was on and decrease during the dark cycle. The dark container DO levels decreased during the entire experiment, and the control remained constant. There was nothing extremely interesting in the trend of pH. The dark container just fluctuated during the 24-hour experiment. For phosphate levels, the trends show the dark and control containers decreased and the light containers increased. Similarly, nitrate levels from the light container increased drastically over 24 hours, and the dark container decreased in nitrate value. The results from the pilot study support the statement that Cassiopeia are able to alter their environment and influence nutrient flux since the treatment tanks differed from the control. As anticipated, for the effective photoperiod study, dissolved oxygen and pH levels decreased in the dark conditions and increased in the light conditions. Additionally, nitrate and phosphate levels increased in the light container, decreased in the dark over 24 hours. The experimental setup we used was found to be a reliable method of continuous monitoring of dissolved oxygen and pH.